Good morning, everyone. Good morning. That's the way to start out a session. Hi, everyone. My name is Liz Watkins, and I am the Dean of the Graduate Division and Vice Chancellor for Student Academic Affairs at UCSF, and I'll be emceeing this morning's session of Discovery Talks. Thank you all for being here. I'm really excited for you to hear this year's lineup of speakers, so my introductory remarks are going to be brief. The Discovery Talks session is the Graduate Division's signature event at this all-alumni reunion weekend, and it gives me, as Dean, the opportunity to show off to you, alumni and friends, the faculty members who are doing some of the most exciting research here at UCSF right now. It's the kind of research that allows us to attract the very best PhD students and postdocs to come here to work with our talented faculty. The mentor-mentee relationship is a mutually beneficial one. Graduate students and postdocs get to work side by side with world-class scientists and scholars, and in turn, those scientists and scholars get the benefits of a highly skilled team to help them carry out their research. We like to say that PhD students and postdocs are the engines that drive the research enterprise, contributing to the discovery and innovation that are the hallmarks of UCSF. And so what you're going to hear today demonstrates the breadth of faculty research taking place at the forefront of biomedical inquiry, with research on topics that span from the level of molecules and cells to individuals and populations. And I think we got a really nice taste of that this morning when we heard our two faculty members talking about telomeres from the molecular level and at the populational level as well. And so now, without further ado, let's get on with the show. The plan is this. The speakers will make their presentations one after the other with just a short introduction from me in between each talk, and you can read more about them in the program. I'm going to ask you to hold your questions until the very end. The speakers have all agreed to stay around afterwards, and they'd be happy to chat with you informally after the talks. So, our very first speaker today is Dr. Anita Cell. Anita is professor in the Department of Microbiology and Immunology in the School of Medicine. She's the co-director of the Integrated Microbiology Program, as well as the Associate Director of the Biomedical Sciences PhD Program, and the Associate Director of the Medical Scientist Training Program. She is an alumna of the Medical Scientist Training Program, having graduated from UCSF with a PhD from the Tetrad Program and an MD from the School of Medicine. Her research is focused on the basic biology of fungal pathogens that cause disease in humans, and the title of her talk is Critical Transitions in Life. Anita.
So I called this talk Critical Transitions in Life because I'm going to tell you a little bit about important developmental changes in a fungal pathogen. But before I do that, I have a confession to make, and it's that I am enamored of fungi. So we share this planet with over 5 million species of fungi, and they're everywhere. So they're part of the normal human microbiome, so they live on our skin, they live in the gastrointestinal tract. They also devastate plant populations in the world. So rice blast fungus, Magnaporta grisei, kills 30% of the rice crop in the world annually. And some of you may have heard about the Dutch elm disease fungus, Ophistoma, which decimated elm trees in Europe, North America, and other parts of the world. On the other side of the story, fungi are a rich source of pharmaceuticals. So probably the most famous example of that is penicillin, discovered by Alexander Fleming, which of course has transformed the way we treat infectious disease. And then other agents like the immunosuppressant cyclosporin, as well as statins, are all fungal natural products. So from moldy oranges in your kitchen to mold in the shower, bread, beer, mushrooms, therapeutics, and disease, fungi really impact many areas of human life. So in my lab, we study a particular type of fungus. We study a number of different organisms. They all are primary pathogens, which means that they cause disease in healthy people as well as immunocompromised people. And these are all organisms that are endemic in different parts of the world in the soil, and they infect high percentages of people who either live in those areas or travel to those areas. And what that means is that in order to study them in the laboratory, we need biocontainment, because obviously we have to protect ourselves, our students, our postdocs, all our trainees from infection. And so we're very lucky at UCSF to have a biocontainment facility that lets us work with these organisms. But that's part of the reason their rich and intricate biology is really understudied. And I want to give you a little bit of the flavor of that today. So the organism that we've studied the longest is one that you can see on the slides, and it's the fungus Histoplasma capsulatum. And it's found in continents, every continent in the world except for Antarctica. And in the US, it's endemic in the Ohio and Mississippi River valleys down into Texas, where it causes upwards of 250,000 infections every year, 25,000 of which are really severe and life-threatening. So this organism grows in the soil, and on the top left of the slide, you can see the soil form of the organism. And in the soil, it grows as a multicellular branched form. I'm doing the interpretive dance in case you can't see the slide from where you're sitting. But it grows as long filaments that branch out and allow the organism to investigate areas of the soil looking for nutrients. But when the soil is disrupted, fragments of those long filaments and spores that are associated with them can get inhaled. And inside the human body, they switch to a unicellular form. And that form is responding, so essentially the fungus is responding to the temperature of the environment. And this is one of the reasons that I decided to study these organisms. The question of how is this microorganism responding to temperature, such a key characteristic of mammals and humans, and changing its growth program entirely, and turning on genes, not only changing its shape, but turning on genes in a program that allows it to cause disease. And you can see that unicellular form on the top right of the slide. Now, for Histoplasma, what that host form of the organism does is it actually infects immune cells. So in the bottom part of the slide, you can see macrophages that have been infected with Histoplasma. And macrophages are immune cells that normally take up microbes and kill them using a large arsenal of antimicrobial effectors. So Histoplasma, in contrast, uses the macrophage as a comfortable home where it can hide out from the rest of the immune system and replicate at very high levels, very robustly. And then the fungus will lyse out of the macrophage, exit the host cell, infect naive macrophages, and start that cycle all over again. So we're interested in all of these processes and the ability of the fungus to manipulate the human host. So one of the approaches we use commonly in the lab is genetics. So we screen for mutants that are defective in various processes. So for example, one of the key things that we've done is identify mutants that are trapped in either the soil form or the host form, independent of temperature. So these are mutants that are defective in genes that are required for the organism to either sense the temperature of its environment or to respond to that signal. And through these means, we have defined a number of molecules and pathways that are critical for the fungus to be able to grow in either the soil form or the host form. And this has been everything from molecules on the surface of the cell to the signal transduction components that are required for that switch, to the transcription factors that drive either the soil program or the host program in terms of causing disease. So we're at a very interesting stage right now of this work, just seeing that the two programs, the soil program and the host program, actually oppose each other. So they're mutually antagonistic, and they negatively, each inhibits the other. And what that tells us is that temperature somehow toggles that switch. So when the organism is in the soil, somehow the temperature of the environment allows that part of the pathway to win out. When the organism finds itself inside a mammalian host, the temperature toggles the switch in that direction, and the host program wins out. And we're now at the stage where we can understand the actual molecular implications, the molecules that are regulated by temperature, in order to allow the fungus to make the appropriate developmental switch. Similarly, in terms of the lifestyle of the fungus in the macrophage, we've identified, using genetics, proteins that the fungus makes, secretes from the fungal cell into the host cell to trigger cell death. And not only are we now able to understand the mechanism of these critical virulence components, but we are also curious about whether these potent molecules might be harnessed in the future for some sort of immunomodulatory function. So I'm going to finish just by hoping that I've infected you with a little bit of my fascination for fungi, and just by I'm hoping that you leave with also this idea of how these understudied, unappreciated organisms really can affect critical transitions in human life. Thanks.
Thank you, Anita. Our next speaker is Dr. Michelle Arkin. Michelle is a faculty member in the School of Pharmacy, and she leads the Small Molecule Discovery Center. She's also affiliated with the Helen Diller Comprehensive Cancer Institute and the Buck Institute for Research on Aging, and she's affiliated with 2 graduate programs, the PhD program in Chemistry and Chemical Biology and the PhD program in Pharmaceutical Sciences and Pharmacogenomics. Her research is primarily focused on developing prototype drugs for new and so-called undruggable targets in cancer and neurodegeneration. And the title of her talk today is, Why is Drug Discovery So Hard, but oh so fascinating? Michelle.
What a cool job I have. Okay, I have a very cool job. Sorry, the mic just went on. So today I want to talk about why drug discovery is so hard, but oh so fascinating. So what is drug discovery? When I'm talking about drug discovery, I'm really talking about small molecule drug discovery, by which I mean one protein, one drug binding to this protein at one site and to nowhere else really in the whole body. Okay, so that's one reason, that's really why it's so hard, is the specificity, choosing the right protein and choosing the right molecule. Other than that, it's easy. So if you look on the left, you'll see the set of chevrons in the funnel, what we call a drug discovery funnel. And you'll see that there's 90% failure at the discovery part, which ends at IND-enabling studies. It starts with selecting a target, ends with safety studies called investigational new drug experiments, and then drug development also has 90% failure. So 1% of the time, when all the smart, well-heeled, well-funded people at a pharmaceutical company decide they're going to invest in drug discovery, 1% of the time that ends up in a drug. Okay? So that's why drugs are so expensive, or one reason they're so expensive. And when we take that 1%, so when you hear numbers thrown around like a drug takes $3 billion to develop or $14 billion to develop, it's not that the drug takes that much to develop. It's that amortized with all the failure and all the efforts that go into the drug discovery engine, you take the expenses divided by the number of drugs, and that's what gives you the number $3 to $10 billion. So if we want to make drugs more plentiful, more affordable, then more in alignment with personalized medicine or precision medicine, then we need to widen that funnel. We need to have less failure. So where do we have most of the failure? Most of the failure in drug development is in the phase 2 clinical trial, so P2 on that chevron. So what happens during drug discovery is that these different stages from target discovery, hit ID, and so forth, we're increasing the complexity of the assays. This molecule starts off just having to bind to that target, but then through this increasing series of more and more complex hurdles, we get to something that binds selectively, doesn't get seen by the body as a poison and then excreted, or as food and then get eaten, but it makes it through to the site of action. That's discovery, so that set of hurdles. And then development is when we're testing that hypothesis, that molecule against that target in humans. And in phase 2 clinical trials, that's the first time we're really testing our therapeutic hypothesis. We're seeing whether that molecule that hits that biology affects that disease in an important way. In some indications, we have 100% failure at that stage. Right? In neurodegenerative diseases, for example, essentially 100% failure. Okay? So what can we do now in the early stages of drug discovery? What we do at UCSF, discover new targets, discover new molecules, prototype drugs, to make that process less failure or more failure proof in phase 2 clinical trials. Okay? So there are three things that I think of as being key challenges that we can address. One is selecting the right target. So why don't we select the right target or the right protein to hit? One, biology is really complicated, and we don't understand it well enough. So if we can take a hypothesis and develop a prototype drug for that, we can test it in a series of animal models, a series of disease models, to reduce the risk or increase the validation that that biological hypothesis will bear out in phase 2 clinical trials. Okay? So that's one thing. The other part of selecting the right target is that sometimes you do know what the right target is. The geneticist, the biologist, the clinician come to me and say, this is the target. And sometimes I have to say, I don't know how to make a molecule that binds to that target. That target to me looks like a sheer rock face, and I can't get a molecule to stick to it. Okay? So when we talk about undruggable targets, we talk about targets that have not yet been drugged. And what technologies, what do we know about proteins or about biology that will allow us to target those proteins with drugs for the very first time? So that's the target. The second thing is that we often don't know the right assay. We don't understand the biology well enough to develop the right assays. Now, at the very earliest stages of drug discovery, we might be testing hundreds of thousands of molecules to see if they have an effect on that biology, maybe 1 million molecules. And so to develop an assay that works at that level of robustness and that level of simplicity that's required to do it 1 million times, we sometimes simplify the system too much, and we simplify the relevance, the biological relevance, out of our assay. So another thing that we try to do in our lab when we're confronted with new biology, new biological formats, is to say, what's the simplest format that I can use that will recapitulate all the relevance and what we think at least is important in that biology? Because we're going to get what we screen for, so we want to make sure that the assay we're performing is biologically relevant. Then the third thing is maybe a more subtle effect. It's this idea of therapeutic index. So therapeutic index, in drug discovery, often we're thinking about activity. What's the concentration? How much protein do we have to have in the blood to get activity? And then at the very end, down when we're thinking about development and we have tested hundreds of thousands of molecules, we say, oh, and is it safe? Is it safe at a dose? Where's the toxic dose relative to the active dose? And that gap, that distance, is called the therapeutic index. So what we want to do also is think about these two complex problems much earlier in the design of a new drug. So how can we do that? How can we both think of all the complexity that is efficacy and activity, and also all the complexity that is safety? Okay? How can we get a safe drug? So to take all of those two complex situations and look at them at the same time, we've turned to supercomputing, high-performance computing, and artificial intelligence that can take all of that information from clinical data, from animal models, from other programs, and merge that together to give us a hypothesis, to give us a prediction of therapeutic index very early in the process. So this is where we are in small molecule drug discovery. Give me a time. How much time do I have? 1 minute? This is where we are in the process. And the reason that drug discovery is so hard is because it's complex. And the reason it's so fascinating is that it's the ultimate problem in systems biology. Because everything that's going to happen to that drug, from what proteins it's going to bind to, whether it's going to be seen by the liver as a poison or food, whether it's going to combine well with the other drugs you're taking, how big and how tasty is the pill, all of that is encoded in 30 to 40 atoms. That is this small molecule drug. So appreciate the small molecule drugs that you have. It's amazing that they work at all. And thank you very much for your attention.
Thank you, Michelle. Next up, we have Dr. Wendy Maxx. Wendy is a professor of health economics in the School of Nursing's Department of Social and Behavioral Sciences, and she's the director of UCSF's Institute on Health and Aging. She's also affiliated with the Health Policy Nursing PhD program, the Center for Tobacco Control Research and Education, and the UCSF Cancer Center. Her research is focused primarily on the economics of tobacco-related illness and tobacco control, and the title of her talk today is The Tobacco Epidemic, Will It Ever End? Wendy.
Thank you, Liz, and good morning. In 1964, the Surgeon General of the United States came out with a report on smoking and health, which definitively established the link between cigarette smoking and lung cancer. At the time of this report, half of all adult men and a third of all women in the US smoked cigarettes. In the ensuing years, there have been a number of tobacco control policies implemented, and we've been somewhat successful. The smoking prevalence has gradually declined. Now, roughly 15% of adults in the US smoke cigarettes, and in California it's even lower. It's somewhere between 10 and 11% perhaps. So we have this tobacco epidemic under control, right? Wrong. About a decade ago, a new product came onto the market, and that's electronic cigarettes, and it really changed everything. Electronic cigarettes, I think I have the middle picture of this, of my slide, shows the evolution of electronic cigarettes. The first generation of the product were these disposable cig-alikes, they were called. And then if you look, the product evolved until now we're on the third and fourth generation. We have tank models that are reusable. You fill them with the e-liquid of your choice. But basically, an electronic cigarette heats a liquid and creates a vapor, and that's what you inhale. As I mentioned, you use an e-liquid in these devices, and these e-liquids come in literally thousands of flavors. I think something like 15,000 flavors have been counted of e-liquids. And that's really important because kids like flavors. We know that of the kids that use tobacco now, I think 80% of them say that they started first with a flavored tobacco product. The newest product on the market that you may have heard of is Juul. And I have a picture in the lower left-hand corner. Juul is a device that looks like a flash drive. You can charge it on your computer. When the devices first came out, kids were using them in their classroom, and the teachers had no clue. The teachers thought they were downloading data or saving files, when in fact they were being used for, they were using them for vaping. Electronic cigarettes have really divided the tobacco control community. Proponents argue that they're a smoking cessation device and that they are safer than combustible cigarettes. It is true that some folks are able to quit smoking by switching to electronic devices, but it's also true that others end up using both. So they try to quit cigarettes and they end up smoking both cigarettes and e-cigarettes. Whether they are safer than cigarettes, we don't really know. Most people would agree that they are less harmful than cigarettes, but they're not harmless. And we're just beginning to understand the impact of electronic cigarette use on health. Remember that smokers don't show all of the negative health impacts of smoking, sometimes for 30, 40 years. E-cigarettes have only been around for a few years, so we're going to be learning more and more as time goes on. But one thing that is clear is that young people are taking up e-cigarette use in alarming numbers. Probably since I started talking, another 500 kids started using electronic cigarettes. The graph on the left shows that among high school students now, about 8% of them smoke cigarettes, but fully 21% report that they vape. So these numbers are really alarming, the way that things have turned around. And the question then becomes, is the benefit to adults who can use electronic cigarette products to cut back on cigarette smoking, maybe they're using a product that has different health effects, I wouldn't say it's healthier, is the benefit from that impact greater than the cost of addicting a whole new generation to these very appealing electronic products? And now there's a new kid on the block. Californians legalized recreational marijuana in November of 2016. And that raises all sorts of new questions for tobacco control folks. How is the availability of recreational marijuana going to impact tobacco use? Are kids going to start using marijuana and then end up switching to tobacco products? How should we tax tobacco? And what are the health effects? How should we tax marijuana? And what are the health effects of marijuana? We don't really know that. It's hard to study a product that's illegal, so now we're going to be having access to all kinds of new data. What are the health effects of being exposed to secondhand marijuana, smoke, or vapor? So there are a lot of new questions to be addressed. Well, what does this all have to do with UCSF? I would argue that UCSF is probably the best place in the world to study tobacco and health. We have a large community of researchers and scientists and postdocs and clinicians who are studying the impact of tobacco on health from multiple angles. We have dentists and nurses and pharmacists, pulmonologists, cardiologists, epidemiologists, toxicologists, biostatisticians, even a few economists. My group has been looking at the economic impact, the cost of the health effects of tobacco use for almost 30 years now. First, we developed models looking at the cost of smoking, and then we turned our attention to secondhand smoke. We've estimated healthcare costs for the Medicaid and the Medicare programs. We've looked at costs for vulnerable populations. We've estimated costs for every county in California and every state in the United States. Our work has been used to help implement some of the policies that have led to reductions in tobacco use. Now we're turning our attention to a couple of the new policies in California. Proposition 56, which was passed in 2017, raised the tobacco tax per pack of cigarettes by $2. So we're looking at what the health effects, the healthcare costs attributable to that policy will be. And we're also looking at Tobacco 21, which raised the legal age to purchase tobacco from 18 to 21. And we're turning our attention to some of the newer tobacco products, as well as poly-tobacco use. It is now very common that people use multiple types of tobacco products at the same time. So, where are we with this tobacco epidemic? When's it going to end? There's some encouraging things going on. The FDA is considering banning all flavors in all tobacco products, and that would include menthol. They're also looking at reducing the nicotine content allowed in tobacco products. The city of San Francisco has played a real leadership role here. You may know that the city of San Francisco has banned the sale of all flavored tobacco products, and they're looking at whether to ban the sale of all electronic tobacco products. So there's some good things happening, and I'm hopeful that the next generation of kids won't be tempted with tobacco products and these appealing devices every time they turn around, but it's going to take time. So I think that the tobacco epidemic is certainly going to end, but it won't be in the near future. And that means that people like me are going to have work to do for many years to come. Thank you.
Thank you, Wendy. Our next speaker is Dr. Stefan Hubble. Stefan is a faculty member in the School of Dentistry and the Oral and Craniofacial Sciences PhD graduate program. He's also affiliated with the Bioengineering PhD program. His research is primarily focused on understanding biomineralization in teeth, dental matrix protein assembly and protein-guided mineralization, and structure property relationships in teeth from the nano to the micrometer scale. The title of his talk today is Carious Dentin Remineralization, the Evolution in Conservative Dentistry. Stefan.
Yeah. Thank you very much, Dr. Watkins, for inviting me to speak here, and that your invitation is certainly one of the reasons why I'm standing here. The other reason why I'm standing here happened about 400 million years ago. And that was not by invitation, obviously, but by design. That's when cells figured out that they can reinforce collagen by adding mineral to it, by adding mineral inside collagen fibrils, and therefore reinforce a tissue and make it weight-bearing and hard and stiff. And that's basically the diagram that you see there. These little red blocks in that yellow fibril are apatite minerals that have been introduced into collagen. So collagen was around at that time already, mainly used as a connective tissue or to separate cell structures. And eventually cells figured out how to get the mineral inside. So what is the trick to actually mineralize collagen? Because if you use collagen like from skin or from the cornea or other organs, other connective tissues, and immerse it into calcium and phosphate, that collagen will not mineralize. It will maybe precipitate some mineral on the surface, but it won't reinforce that fibril. So what's so unique in getting the mineral inside, and the step in evolution was basically the development of phosphorylation of proteins in the extracellular matrix. That phosphate group that usually sits on serine attracts calcium. The calcium in return attracts more phosphate, and you sort of build up a molecule that is surrounded by a cloud of calcium and phosphate. If that cloud attaches to collagen, the ions are then released inside the fibril, as you can see on the bottom graph there, and gradually form an amorphous calcium phosphate. So a mineral develops, and over the period of a few days, that amorphous phase will turn into crystals, like apatite crystals that are nicely oriented with the collagen molecules within a fibril. So that way you reinforce a structure that is very pliable. The collagen fibril of skin is very easy to deform. It's more like a well-cooked spaghetti, and you transform it into something hard and stiff by adding tiny little apatite crystals of the size of about 20 nanometers down to a thickness of only 2-3 nanometers. So it's sort of a very unique nanotechnology these cells have developed in incorporating mineral into an organic structure. So that is done by these phosphorylated proteins like dentin matrix protein, osteocalcin, osteopontin, the sialo proteins. Those can organize the mineralization, but those are very scarce protein. Phosphorylation cannot be done by bacterial cells, so recombination is not an option to make these phosphorylated proteins. So it would be great to have a synthetic substitute so we could mimic mineralization of collagen in vitro and also make mineralized tissues by just simply using collagenous matrices like reconstituted collagen and add a mineralizing agent and turn it into something like bone or dentin. Or maybe turn around a process like caries, which is based initially at least on demineralization, and reverse it and remineralize the tooth again and therefore repair or heal an actual carious lesion. So that molecule then was actually discovered about 15 years ago, and it's quite simple. It's actually a polymer, and it's called polyaspartic acid. So it's actually aspartic acid, like the residues, that's charged with a carboxyl group on the outside, just like negatively charged like phosphate. It's all aspartic acid, so it's about 20 residues of aspartic acid. It attracts calcium. Calcium attracts the phosphate. It makes this ion cloud surrounding polyaspartic acid. When you deliver polyaspartic acid in calcium and phosphate to dentin matrices, those matrices start to remineralize. So you can see on the upper left, there's an artificial lesion where we demineralize dentin. We immerse then this structure into a polyaspartic acid solution. And over time, you can see how the mineral recovers. Within 2 weeks, we basically have 100% recovery of the mineral and about 50%, 50 to 70% recovery of the properties like hardness and stiffness. So that's all nice, but this is still a solution. This is just you're dropping your sample, your tooth, inside a beaker. How would you apply that in dentistry? Obviously, you can't treat patient and have them gargle for days, basically, in a calcium phosphate solution. So we worked on developing a method how to include the release of polyaspartic acid within a restorative treatment. So we have developed cements. These are glass ionomer cements that actually use polyaspartic acid for the setting reaction. So we it's very simple. We mix polyaspartic acid with bioglass, and these two react with each other in an acid-base reaction and set as a cement. And you see basically on the bottom left how we applied that cement to a carious lesion and then did a micro-CT over time. And you can see that the lesion starts to recover within about a month. So we have now just lately received data where 50% of the lesion are remineralized within only 4 weeks of treatment. So of course, in a patient, you would apply that restorative, and that would remain as long as the restorative lasts. So it could be actually months or years of release of polyaspartic acid that will encourage the repair and the remineralization of the damaged collagen. That approach also implicates that you cannot treat caries as we used to do in dentistry, where you have a fairly small lesion, well, like on the upper right, and with such a lesion, the dentist would remove the entire lesion, obviously, as they were trained, but also remove a large portion of the healthy and intact tissue just to conform with the right geometry of the restoration. So in these new approaches, we actually combine our remin strategy with minimal invasive dentistry, where you now remove only the infected portion of the lesion, which involves the enamel and a portion maybe of the dentin, then apply the remineralizing cement, our PILP cement, and then restore just like you usually do with a composite or with a cement, and then have the tissue heal underneath. That way, you have conserved a large amount of the healthy tissue. We actually were able to repair or heal lots of the damaged tissue. Therefore, you're less likely to, you basically extended the lifetime of the tooth, and you're less likely to require any endodontic treatments, because even if there's a second treatment that often happens with restoration, or a third, there's more tissue left behind that the dentist can now treat again, and therefore you're less likely to require endodontic, a root canal treatment eventually, or the loss of a tooth entirely. So as we are approaching now lunch more and more, caries obviously is still an issue, and many of you, I'm sure, had a lesion in their lifetime. So brushing and maybe less sugar is definitely beneficial, and hopefully you will never have to go and see the dentist. But in case you have to, dentistry is certainly changing, and the approaches on how we treat teeth become less invasive, more conservative, and if they include these remineralizing agents, we will also have a method how to heal at least a portion of the lesion and conserve your tooth, hopefully for a long time. So thank you very much.
I am really excited about our fifth presenter. So we found out at the very last minute that unfortunately Dr. Dyke Mullins is going to be unable to participate today. He was unable to attend. Very fortunately, we have with us this year's winner of the Postdoc Slam. Let me tell you a little bit of what that's about. In the graduate division, we think it is vitally important for our trainees to learn to communicate complex scientific concepts and research to a non-specialized audience, much like yourselves and much like the 4 faculty members have just done, taken their very complicated, specified research and presented in a way that we could all understand and appreciate. So 5 years ago, we started a competition for graduate students called Grad Slam. This is where students compete to present their dissertation research in 3 minutes or less to a non-specialized audience. We have a big live finals. We have 10 finalists. We don't have a football game or football team at UCSF, but it's the closest thing as you can get to a football game. It's a little bit like American Idol for scientists, if you can imagine. We fill the auditorium. People come and cheer for their favorite scientists. And we have a competition, and we give a first place award. First place, second place, third place, and we do people's choice. 3 years ago, we realized that we should do this for our postdocs as well, that our postdoctoral trainees were at a slightly more advanced stage, becoming closer to becoming faculty or to whatever career they were going into, and they too should be able to communicate their research in 3 minutes or less. This year's winner is Sona Chaudhary, and she is going to be presenting at the Graduate Division Gala tonight, but she very graciously agreed to step in and pinch hit. So let me tell you a little bit about Sona, and then you're going to see her prize-winning speech. Sona is a postdoc in Dr. Joel Palevsky's lab in the School of Medicine. The title of her talk is the Butt Cancer That No One Talks About, and I'm going to let Sona tell you where that title comes from. But please give her a warm, warm welcome for stepping in at the last minute.
Hello, everyone. So I need you all to imagine I don't have my slide here, and the music is not there, but the music is Shake Your Booty. So that's the music. I'm just setting the mood. Valentine's Day 2018 will always be special for me. Can you guess why? I received a rare gift, the first sample of human butt tissue from a patient undergoing surgery for anal cancer. Anal cancer is linked to a sexually transmitted virus called human papilloma virus, or HPV, the same virus that causes cervical cancer. HPV infects the skin of the anus and interferes with the normal working of the cells. These cells may become precancerous and ultimately form a tumor. Now, if we can catch the disease at the precancerous stage, we may have a chance to prevent anal cancer. My research focuses on identifying a marker in the precancerous anal tissue that will help in early cancer detection and treatment. The marker is a genetic material similar to DNA called microRNA. These microRNAs are abundant in our cells, and they play an important role in controlling a number of biological functions. So what is the link between HPV, cancer, and microRNA? Imagine the microRNA as a music conductor orchestrating the genes so that they work in perfect harmony. However, infection with HPV heckles the conductor, and he misses the note, and he loses control of the genes. And the result is cancer. I have succeeded in identifying microRNAs in anal tissue by dissecting out the disease region with a laser and then extracting microRNAs. After complex data analysis, I found that the quantity of a particular microRNA, microRNA 34C, was far less in precancerous tissue compared to normal tissue. So my hypothesis is when this vital microRNA is depleted in HPV-infected cells, it pushes the cells towards a precancerous stage. I am currently investigating whether we can reverse anal cancer progression by providing this missing microRNA back into the tissue. If successful, I envision that one day we can provide these patients with a therapeutic topical cream containing the missing microRNA 34C that potentially can prevent anal cancer. I know it's awkward to talk about what's going on in the butt. But to prevent anal cancer, we need to talk about the butt. Thank you.
Thank you, Sona. I think you can see why she was the judge's top choice, and she also won the audience top choice award. Thank you, Sona. Thanks to our speakers. Can you join me in another round of applause for all 5 of our speakers? I hope this has just given you a little taste of the exciting research that's being done in the labs and research groups across UCSF at all levels, from professors to our trainees. That's our show. The speakers have agreed to stay around for a little while if you have questions you'd like to ask them. I hope you enjoy the rest of the weekend. Thank you for being here.